Hello to all students. I hope you are doing fine. In this video lecture, we are going to discuss color blindness and genetics of color blindness. First of all, I am going to briefly describe the structure of human eye. As you know that our eye consists of three layers. The innermost layer is known as retina. The innermost layer of retina is responsible for vision because this retina, innermost layer of the eye, contains specialized cells which are known as photoreceptors. Photoreceptor cells are of two types. One of them are called rods while other are called cones. Rods have special types of cells which contain a protein known as rhodopsin, which is responsible for in vision of dim light, while the cone cells contain a specialized protein which is known as opsin, which are responsible for vision in bright light. So basically the cone cells are responsible for color vision. So there are three types of basic colors and there are three types of basic cone cells: blue cone cells, red cone cells and green cone cells, depending upon the type of the color. So they help to identify different shades of these primary colors: blue, green and red. So when there is a problem, usually genetic problem, when these cone cells are not properly functioning, we are unable to see colors or their shades. So that is why a problem arises in human beings that is known as color blindness. So there are different types of color blindness. Let's move on to the different types of color blindness. First of all, I am going to discuss dichromacy, which is a type of color blindness, which is a hereditary disease. It is sex-linked trait, usually X-linked trait, because its genes are present on X chromosome. And there are three types of dichromacy. First of all, I am going to discuss protanopia. Protanopia is a red blindness. In this condition, red cone cells of retina, they do not function properly. So that is why the person affected from this type of color blindness like protanopia unable to distinguish red colors and shades of red colors. So they will see red as black or gray in color. So that is a very big issue for any male or female who suffers from this type of color blindness. The second type of dichromacy is Deuteronopia. In Deuteronopia, this is known as green blindness. The proteins opsins present in green cone cells are unable to distinguish green colors, and the person who is suffering from Deuteronopia unable to see green and all the shades of green colors, and they usually see as a black or gray color of green shade. The third type of dichromacy is Trichromacy. Trichromacy is blue blindness, and in this case blue cones of the retina are not functioning properly and the person affecting from this color blindness unable to see blue color, and usually the blue color is seen as greenish shades. So these types of color blindness is not usually a big problem. The actual color blindness is monochromacy, which is a true blindness. Monochromacy is total color blindness or actual color blindness. It has two types. Number one, rod monochromacy. Rod monochromacy, in this condition of color blindness, people are unable to visualize any color. So there is no colorization in this disease. And all the things you see or a person affected from this type of color blindness see the things as black and white, just like a black and white TV. The second type of monochromacy is cone monochromacy. In this condition, two dichromacies join together, like protanopia and deuteranopia join together. Protanopia and trichromacy can join together. Trichromacy and deuteranopia can join together. So in this way, there are two types of dichromacy. So double dichromacy this is called, and the vision is normal In this condition, vision is poor, but in this condition, vision is normal, but the person affecting from this disease unable to see two different colors. For example, the person suffering from cone monochromacy are unable to see red or green, green and blue, blue and red. Any two colors cannot be distinguished properly. So those persons are real risk of color blindness, and these are known as monochromatic. The third type of color blindness is trichromacy. Trichromacy is known as altered color vision. In this condition, the person affecting from this disease can see color, can perceive colors, but with altered perception, with altered shades, not actual shades of the colors. So there are three types of trichromacy. Protonomaly, usually the person sees shades of red and green which are mixed with each other, red and green hue. And the second type is Deuteronomy, which is usually in the person same condition like protonomaly. In this condition, red and green hue, the person see these colors but with different or altered shades. The third type of this is known as Deuteronomy. This is blue-green trichromacy. In this condition, the person affecting from color blindness can see shades of blue and green, but they are mixed with each other with altered perception. So now we are going to discuss how this disease transferred from one generation to another generation, because it is a hereditary disease and runs in families. So we are going to discuss its genetics. So first of all, I am going to make a cross between a normal mother and colorblind father. Female, she is normal, while father, male, is colorblind. For normal mother, for normal female, there are two X chromosomes present inside the normal mothers only. So we are going, because that is a X-linked trait or sex-linked trait, so we are only discussing sex chromosomes in this cross. So there are two X chromosomes present in normal mother. X chromosome does not carry anything on it, so that is why this person is normal. While colorblind father is XY, and its X chromosome contain a gene which is represented by letter C. So it means that this father is colorblind. When they got married, mother produces gametes which are eggs. Father produces gametes which are sperms. During meiosis, during formation of sperms and eggs or gamete formation, meiosis takes place and these chromosomes get separated. In each egg, one X chromosome is present, and in each sperm, one X and one Y chromosome is present. At the time of fertilization, there is a random fertilization. Any egg can fuse with any sperm, or any sperm can fuse with any egg. So there are some probabilities. So in the F1 generation, all these are the progeny or offsprings. There are 4 offspring from this couple, and there are 2 daughters and there are 2 sons. If you look at the genotype of a daughter, this daughter received one X chromosome from mother, which is normal, and she received X chromosome from father, which is colorblind. She is normal, but at this stage, because she is carrying a gene for color blindness, so that is why she is carrier. So we call this a carrier daughter. In the same way, the second daughter is also carrier, because she takes normal gene from mother and colorblind gene from father. While the sons are also normal, because a son inherit X chromosome from mother and Y chromosome from father. So both of the chromosomes are normal. So in this case, both of the sons are normal. So in this way, the father, which is colorblind, has transferred or inherited its genes of colorblindness to his daughters, not to his sons. Now I am going to move into next cross. In the next cross, I am going to cross between this carrier daughter with a normal father. So I am going to take this carrier daughter right over here. This is a carrier mother now, and the father is normal also, and they both got married. During gamete formation, eggs are produced. One egg is with colorblind chromosome and one egg is with normal chromosome. It's in the same way, the male sperms are both carrying normal chromosomes. In the progeny, in the F2 generation, there are 4 offsprings. First daughter, she received X chromosome from mother and normal X chromosome from father. She is carrier daughter. Second daughter, she received normal chromosome from mother and normal chromosome from father. She is normal female. She is also normal, but she is carrier. In the same way, this son, which is a male, she received colorblind X chromosome from mother and Y chromosome from father, and he becomes colorblind. He is affected, while the second son is normal. So, in this way, uh, these are the offspring of these father and mother. So, this mother is taken from this progeny. So these are the progeny offsprings. So this is the mother. So the mother has transferred its colorblind gene to his son. And if you go back to the cross, so this daughter, which is the mother right over here, has received this chromosome, this gene of color blindness from her father. So this gene moves in a zigzag fashion. It moves from father to his daughter, and from her daughter to his mother, and from daughter to her son. So in this way, this son, which is a colorblind male, has received its colorblind gene from his maternal grandfather. So now let's move on to the one more time and one more cross to understand more deeply about this genetics. So now I'm going to marry this colorblind male with the carrier mother, or this carrier female to a colorblind male. So I have taken another cross right over here. These are P3 carrier mother. She is taken from right over here, and colorblind father, which is taken from right over here or from right over here. So in this cross, you can see that there are two types of eggs are produced, one with colorblind, one with normal chromosome. It's in the same way, father produced two types of sperms, one with colorblind X chromosome and the second Y normal chromosome. This is their progeny, offsprings. The first daughter, she received X chromosome, which is colorblind, from her mother, and she received X chromosome from her father, which is also colorblind. So when both of the chromosomes appear in the same female, then she will become colorblind. So in this way, usually this disease is present mostly in males, because males only have single X chromosome. So if any of the gene is present, if there is a colorblindness gene present, they will become colorblind. And for the female, it needs two chromosomes, two genes to become colorblindness. So now you can understand this genetics right over here. Second daughter, she's a carrier female because she received normal gene from mother and colorblind gene from father. In the same way, one son, which is colorblind, and one son is normal, you can understand from this cross. So in this way, this disease is usually mostly present in males, and in the less amount it present in female. Anyhow, if this disease is present in male, it's very problem. They cannot drive, they cannot fly aeroplanes, they cannot go to army, they cannot distinguish colors, but their problem can be solved, or there is no treatment for this disease, but it can be enhanced by lenses or glasses. But this problem, especially for the female, is very severe, because the female will be unable to distinguish different types of colors of their clothes, makeup, cooking, and other problems they can arise in their life, and life become a little bit harder. But new technology can make little bit easy for those people who are suffering from color blindness. So I hope it makes sense, and I'll see you in the next lecture. Until then, bye.